mosquitoes are the worst. The buzz sound and their thirst for blood is just plain annoying. You just can't sleep peacefully at night, especially when they are associated with a number of life-threatening diseases. Dengue is a mosquito-borne disease that is dramatically growing around the world and is still continuing to instill a public health concern among the affected countries. The number of dengue cases reported to the World Health Organization has increased 8-fold over the last 2 decades. A modeling estimate indicated that 390 million people succumb to dengue virus infection annually. With dengue being endemic in more than 100 countries, WHO revealed that 70% of the actual burden is in Asia. Ever since, conscientious endeavor for drugs and vaccines to treat dengue are rapidly entering clinical trials. However, most of them failed the clinical trials due to the presence of side effects or the lack of efficacy. Till date, no specific dengue drug or vaccine can confer protection against the virus. Dengue vaccine is an exception. Nevertheless, it only serves as a prophylaxis for patients with a history of dengue infection. Ultimately, this led us to the project of in silico design and analysis of dengue peptide inhibitors. The primary aim is to inhibit the interaction of dengue non-structural proteins with their respective receptors and disrupt viral life cycle. Dengue is a single positive-stranded RNA virus from the Flaviviridae family. Its genome alone consists of 3 structural proteins and 7 non-structural proteins. In this study, we decided to narrow down to 3 non-structural proteins. NS1 is a glycoprotein and the only protein to be secreted out of the infected cell. This protein is known to cause dengue hemorrhagic fever and dengue shock syndrome. NS2B, NS3 on the other hand, is a serine protease cofactor that cleaves polyproteins to separate different dengue proteins. Finally, NS5, the largest protein with a RNA-dependent RNA polymerase and a methyltransferase domain, is essential for viral replication and primarily relies upon the human signal transducer and activator of transcription degradation to evade the host's immune response. The drug development process itself is laborious and costly. However, the benefit of approaching such problems computationally not only is cost-effective and time-saving, it also paves way to efficient clinical-ready pharmaceuticals. Rational design strategies using prior knowledge or computation to maximize target affinity and bioavailability have given rise to some successes. But why peptide inhibitors? Firstly, peptides have a low toxicity and a high specificity towards their target. Peptides can also be easily synthesized, and its biosynthesis reduces environmental pollution and energy consumption as opposed to the synthesis of small compounds, something stakeholders would very much benefit from. Recent reports envisage that the peptide market will drastically increase in demand by the next decade, with North America expected to dominate the market together with other major pharmaceutical companies. With the market being moderately competitive as of now, we believe peptide therapeutics will be a future asset to the stakeholders of the industry. The NS5 viral protein is a dimeric protein and is the largest in the dengue genome with a molecular weight of 103 kilodalton. Its overall structure is 900 amino acids long. The composition of NS5 is also well conserved across its family, consisting of 2 domains, which are the methyltransferase domain and the RDRP domain at N-terminal and C-terminal respectively. The latter encompasses 2/3 of the protein compulsory for the generation of infectious virions. The NS5 protein has been considered a vital drug target due to its role in spearheading dengue viral replication. STAT2 is a protein-coding gene crucial in the interferon pathway. However, the dengue NS5 protein interacts with STAT2 and degrades it to evade the host immune system in order to facilitate efficient viral replication. In this study, peptide inhibitor was designed to inhibit this interaction. The objectives of this study were to determine the interaction of the dengue NS5 viral protein and STAT2, to design and establish a library of peptide inhibitors through computational saturated mutagenesis, and to generate the best peptide inhibitor that would competitively bind to dengue NS5 viral protein. The computational pipeline to design dengue NS5 peptide inhibitor involved 5 major steps before concluding with the genetic circuit design. The figure shows the homology models of Dengue NS5 protein monomer and the human signal transducer and activator of transcription, or in short STAT2, which was extracted from the Zika virus RDRP complex. Both protein structures had good stereochemical quality. Based on the Ramachandran plot, 94.3% of NS5 protein amino acid residues were in the favored region, while 2 amino acids which were not crucial in the study were in the disallowed region. 90.2% of STAT2 amino acid residues were in the favored region, while 1 amino acid was found to be in the disallowed region. Docking analysis between both proteins revealed the crucial involvement of coiled-coil domain of STAT2 encompassing amino acids 137 to 316 with both NS5 domains. Subsequently, based on the interaction sites in parallel with experimental reports, few wild-type peptides were selected from the STAT2 protein itself. Upon mutation using an automated pipeline, MutateX employing FoldX, the analysis showed that many mutants had better stability and interaction energy. A negative binding affinity change caused by mutation simply dictates that the mutant amino acid is more stable than the wild type, while a positive value indicates that the amino acid substitution has a destabilizing effect. Interestingly, aspartic acid at position 291 and arginine at position 300 were indispensable for the peptide. This is largely due to the important function of the residue in preserving its integrity of structure and binding interface between Dengue NS5 viral protein and peptide complex. The few best mutant peptides were then selected from each wild-type peptide for further analysis. The mutant peptides were named according to their wild-type origin, position, and amino acid residue before and after substitution to ease the analysis. Reanalyzation of docking performed through the Haddock web server revealed that most mutant peptides showed lower Haddock score compared to the wild-type peptides. However, peptide 1T193F had the lowest and the best Haddock score compared to the rest of the mutant peptides. So, peptide 1T193F was considered for further analysis. It was observed that the peptide formed 2 hydrogen bonds and 23 hydrophobic bonds with the viral protein. The mutant peptide derived from the STAT2 CCT domain was seen to interact with both domains of the NS5 protein, crucial for RNA capping and viral replication.
The structure that you're looking at is the Dengue NS1 protein. The NS1 protein comprises of 352 amino acid residues and shares approximately 70% sequence similarity among all 4 dengue serotypes. This protein is really special in the sense that it does not exist in a fixed size. It comes in various forms such as monomer, dimer, and hexamer, depending upon its glycosylation status. The structure that we are looking at is the dimer form, where 2 monomers come together and interlocks at the beta role. The beta role is also known as the hydrophobic core, and it is the target of our inhibitor peptide design.
Earlier, I mentioned that the beta role is the target for our antiviral peptide design. The reason for this is because the beta role is very important for the hexamer formation. You can see in the diagram on the left, the middle region, which is the beta role, is colored yellow, indicating that it's the hydrophobic region. This hydrophobic region comes into contact with lipid molecules. When 3-dimer NS1 comes into contact with a lipid molecule, they form a hexamer. A hexamer is very important in order for the NS1 to be secreted out of the infected cell in a stable form. And secretion of this NS1 have been known to cause many severe dengue diseases such as the dengue hemorrhagic fever and dengue shock syndrome. And the purpose of this project is to stop the secretion of hexamer. This is the workflow of computational pipeline to design the Dengue NS1 peptide inhibitor. In the first step, the structure of the NS1 was retrieved from RCSB PDB and it was viewed using Chimera. The purpose of viewing using UCSF Chimera was to examine for missing residues, particularly at the beta role region. However, it was found that there were some missing residues, and therefore the next step of homology modeling was performed using YASARA, and it was further validated using IRAD and ProCheck. After that, in step number 3, the peptides to be used for the molecular docking was obtained from ChemPR3 database, and a virtual screening was done using Yasara. In the 4th step, LigPlot Plus was used to examine the interacting residues between the target peptide and the NS1 protein. These interacting residues was further mutated using fold X during the site-directed saturated mutagenesis to improvise the binding energy. And these mutants and the NS1 residues were further examined using LigPlot Plus to examine for the interacting residues. The homology model that was generated using Yasara was further validated for its quality using Ramachandran plot analysis. According to this Ramachandran plot analysis, 87.2% of the residues found in the NS1 protein fell into the favored region of Ramachandran plot. This indicates that the model generated using Yasara was of high quality. The homology model was again validated using ERAT. And this time, both chain A and chain B of NS1 protein scored a quality factor of 92.868. And this indicates that the model generated was again of high quality. 4 alpha helix peptides have been selected to dock against the NS1 protein, particularly at the beta row region. And the peptide with the PDB ID of 4-MGP recorded the highest binding energy, about 9.52 kcl mole. This peptide was then brought into the following step of site-directed saturation mutagenesis. Prior to mutagenesis, the interacting residues between the selected alpha helix peptide and the NS1 protein was studied. The green line indicates the hydrogen bonding, and the red lines are the hydrophobic bonds. And the molecules that you can see, the pink represents the NS1 DENV protein, whereas the red ones are the alpha helix peptide residues. The results that was obtained upon performing the mutagenesis was 10 mutants. Out of these 10 mutants, 3 recorded the highest binding energy, and they are highlighted in blue. And these 3 peptides were brought into further validation using HADOC.
The docking result that was obtained previously was validated using HADOC 2.4 web server. As you can see, compared to the native peptide, the mutants, which are 3 mutants, scored a better HADOC score. This shows that the mutant had better binding affinity towards the NS1 protein compared to the native peptide.
The dengue virus NS2 protein consists of 2 domains, which are the protease domain at the N-terminus and the multifunctional domain of helicase, nucleoside triphosphatase, and 5' terminal RNA transphosphatase at the C-terminus. It is among the most conserved non-structural protein in all 4 serotypes of dengue virus, with the similarities of 77% in the amino acid sequences. Another protein, known as NS2B, is the cofactors of the dengue serine protease. Its association with the NS3 protease domain is essential for the proteolytic activity to be carried out at its full capacity. Due to the importance of proteolytic event in the life cycle of dengue virus, NS2B NS3 is an important target for drug screening in search of antivirals against dengue. Since the viral genome of dengue has only a single open reading frame, all the structures and non-structured proteins of dengue are translated together in the form of polyprotein. For each of these proteins to be functional, they should be separated from each other by proteolytic cleavage at the endoplasmic reticulum membrane. Within the ER elements, the host proteases such as signalase and furin are responsible for the proteolysis of the polyprotein. In the cytoplasm, it is the dengue NS2B NS3 protease that separates these proteins by cutting them at the proteolytic cleavage site. In our drug design, we mimic the sequences of the proteolytic cleavage sites with the aim of developing potent NS2B NS3 inhibitor. Dengue NS2B NS3 protease cleaves the polyproteins at 8 different sites. The cleavage sites of protease are designated to be non-prime and prime sites in order to differentiate the N and C termini respectively. The figure here shows the cleavage sites of the protease with the arrow pointing towards the cis bond. Due to the recognition of the linear peptide sequences as a substrate of the Dengue NS2B NS3 protease, peptide cyclization was taken as a strategy to avoid the degradations of the peptide inhibitors. To achieve peptide cyclization, the sunflower trypsin protease inhibitors, or SFTI1, was chosen as a scaffold due to the previous report of its suitability in peptide inhibitor design. In our study, almost all the prime and non-prime site residues are substituted with the amino acid sequences found in the 8 cleavage sites mentioned earlier, yielding various mutants with different activities against dengue protease. The only exception is the conservation of the P3 residues for all mutants due to the importance of these cysteine residues in the formation of stabilizing disulfide bridge. These P3 residues is highlighted with red circles here. Here is the general workflow of the computational pipeline. The peptide design starts with the structure retrieval and validation using Protein Data Bank or PDB, PDB Redo, and SAFE version 6.0. Homology modeling was then carried out for the missing residues of the crystal structures using Yasara, and then minimized followed by quality check. Docking and mutagenesis of the cyclic peptides were conducted afterwards, giving results that were visualized and analyzed using Zher and LigPlot+. Sequence manipulation suite and the codon optimization tool provided by Integrated DNA Technologies were then used for reverse translation to obtain the DNA sequences of synthetic genes and codon optimized to ensure satisfactory expression in the chassis. This table shows a comparison for the wild-type SFTI1 and its various mutants. The best result of both Yasara's binding energy after minimizations and the Haddock binding energy respectively are ranked first, and the rank goes on. Interestingly, SFTI1 capsids ranked first for both the results, indicating that it is the most stable and most potent peptide inhibitor. This 3D image of the dot complex show that the wild-type SFTI1 is able to bind with the Dengue NS2B NS3 at the active sites, indicating its suitability as the scaffold for peptide design. 2D visualizations of the protein-peptide interaction using DENVer functionalities of the LigPlot+ shows the formations of many hydrophobic interactions, as shown in red, and hydrogen bonds the hydrogen bond shown in green, which again proving the feasibilities of using wild-type SFTI1 for drug design by subsequent mutagenesis. Similarly, this 3D image of the dot complex shows the SFTI1 capsid is able to bind with the Dengue NS2B NS3 at the active sites, showing its inhibitory potentials. The 2D visualizations of the dengue virus NS2B NS3 interactions with STFI1 capsid mutant peptides aligned with its best results of stabilities and binding energy shown in the paper earlier. In this diagram, it is clear that significantly more hydrogen bonds and also the hydrophobic interactions shown in green and red respectively reflects its potency, which needs to be validated using random experiment in the future. This table summarizes the best peptide inhibitor for each viral protein and its DNA sequence. The best inhibitor for NS1, NS2B NS3, and NS5 protein are gaining 2, AU15R, SFTI1 capsid, and peptide 1T, 193F respectively. Excluding the start codons and stop codon, all of them are less than 1 base pair in length, with the GC content between 40 to 60%. Now I'm going to present the ideas of our genetic circuit design. The main ideas of our genetic circuit design revolve around the control of cell growth, IPDG-induced expressions of T7 RNA polymerase and CI repressor, peptide expression and its regulation, as well as cell stress-induced expressions of PET repressor to prevent cytotoxic inclusion bodies accumulation. At the final stage of fermentation, glucose deprivation slices the bacteria to kill them and release the peptide products simultaneously. Control of cell growth enables us to channel nutrients to synthesize desired products such as peptides in our case, instead of wasting it for unwanted biomass production. The conventional approaches to control cell growth are suboptimal as the antibiotics-based approach can kill the chassis, while alternative approach of limiting essential nutrients will affect the yield of our peptides. To optimize our genetic circuit, we decided to use knockout strains of E. coli, which can produce its own beta and beta-pi subunits of RNA polymerase. Under the control of P-Lux promoter, our chassis can only grow upon the additions of HSL, or in its full name, N3-oxohexanyl L-homoserenactone. The only condition where its RNA polymerase can be completely assembled. The solid proof of its feasibility has been reported by Isaac et al., where the controlled expressions of RNA polymerase enabled twofold increase in glycerol productions without killing the cells. Upon reaching the desired populations, the removal of HSL stops the chassis from growing but retains active metabolism for our peptide synthesis. The peptide biosynthesis starts with T7 RNA polymerase expressions, being placed under the tight control of P-LEC-IQ and LEC-UV5 promoter. This protein is only expressed in the presence of inducer IPTG. IPTG is only added when fermentation is intended and no further cell growth is wanted. IPTG is only added when fermentation is intended and no further cell growth is wanted, as mentioned earlier. Under the same promoter, CI repressor is produced as well. This repressor binds with CI-operated upstreams of the RPOB and RPOC genes to block any possible leaky expressions, further strengthening the cell growth control. The P-LEC-IQ used here is stronger than the wild-type P-LEC-I promoter, thereby reducing the leakiness of the downstream expression. LEC-UV, on the other hand, is independent of the catabolite repressions by glucose, enabling T7 RNA polymerase and CI expressions under high concentration of glucose, our carbon source of the fermentation. The T7 RNA polymerase produced earliest binds with the T7 promoter, enabling antiviral peptide expression. When the peptide is overexpressed, inclusion bodies will be formed, which can cause cell stress and cytotoxicity. Hence, we incorporate cell stress-inducible TET-R expression system, making use of the sensitivities of the PIBP-AB promoters to the presence of inclusion bodies. This activates the expressions of TR expressors. This activates the expressions of TET-R repressor. TET-R repressors binds to the TET-O operator upstreams of the peptide genes, thereby inhibiting further overexpressions of peptide. During the production phase of fermentations, high glucose concentrations and the absence of arbinose repress the arbinose promoter, thereby preventing the unintended expressions of choline and endolysin. To harvest our peptides, the carbon composition is reversed with glucose deprivations and presence of arbinose respectively, activating the expressions of cell lysins, cholines, and endolysin. This kills the chassis and releases the peptide products simultaneously. In the Asian pre-COP-MUD biosafety workshops, our team suggested the use of kill switch for biocontainment of GMO and discussed biosafety issues with stakeholders such as industrial players and biosafety regulators. A Japanese scientist made an interesting point that kill switch shouldn't be used, saying that it is dangerous. With the concerns of the stakeholders in mind, we did not proceed with the ideas of including antitoxin systems in our genetic circuit design. Instead of providing antitoxins, we changed it to an arbinose-inducible systems, where the choline and endolysins are only expressed after fulfilling two conditions: the absence of glucose and the presence of arbinose. Our design can address the concerns of mutational escape from kill switch effect, since the leaky expressions of its antitoxins, more specifically anticholine, is impossible. The controls of the choline analyzing systems via glucose deprivations, and arbinose inductions can also fix the initiations of cell lysis to our hand.
The main idea of our genetic circuit design revolves around HSL-based control of cell growth. IPTG-induced expression of T7 RNA polymerase, regulation of peptide expression by T7 RNA polymerase and TET repressor, and cell stress-induced expression of TET repressor to prevent cytotoxic inclusion bodies accumulation. At the final stage of fermentation, glucose deprivation lyses the bacteria to kill them and release the peptide products simultaneously. In order for the viruses to induce a disease, they must first mask themselves from the body's immune system. With the Dengue NS5 targeting the STAT2 for the degradation and disrupting the host interferon signaling pathway, the mutant peptide 1T193F can be a potential peptide therapeutic candidate to competitively inhibit the Dengue NS5 viral protein from binding to STAT2. The alpha helix peptide-based drug design approach was used for designing the NS1 inhibitor peptide, with the intention to create a highly hydrophobic inhibitor peptide compared to the lipid molecule that helps in the formation of the NS1 hexamer. That is why alpha helix are commonly found in the hydrophobic core of the quaternary structure of proteins. In general, the alpha helix is also stable compared to the linear peptide, as it has a stable secondary structure and thus having a low chance of proteolytic degradation. From the work that was done, 3 mutants have been found to bind strongly towards the beta role of the NS1 protein, namely alanine 15 arginine and alanine 15 glutamine and glycine 1 lysine. These mutant peptides could be potential solution to stopping the severe forms of dengue disease caused by the NS1 protein. The mutant peptide known as STFI1 capsid can be a potential peptide to inhibit dengue NS2B NS3 serine protease. In the design of serine protease inhibitor, a slightly different approach has been taken by incorporating peptide cyclization. This is important to prevent the proteolysis which would otherwise happen on the linear peptide. In the future, the cleavage sites of the dengue serine protease at the human proteins such as STING can be considered with the aim of developing potent. Metabolic engineering and genetic code expansion may also be incorporated in the future, where the expression hosts can synthesize and incorporate unnatural amino acids in the peptide inhibitor. Development of therapeutic peptide is no doubt not only full of challenges and risks, but also offers great potential. Rational structure-based design can open up to more powerful insights in designing future generations of peptide drugs with the greatest safety and efficacy. Future works could potentially study to incorporate peptide stapling or cyclization to increase target affinity, cell penetration, and protect against proteolytic degradation.